Good morning. Welcome to day one of our distance learning. I'm going to be walking through chapter 23 notes, just the end of it that we didn't get a chance to cover. So here, I'm going to share my screen with you and walk through them. All right. So we've talked about Hardy-Weinberg. We talked about the 5 parts of Hardy-Weinberg equilibrium. We talked about how most of the time we don't have that. So evolution really is looking at those 5 agents of change that we talked about last week. So we're looking at small population size, non-random mating, mutations can occur. The other two, genetic drift and gene flow. So today we're going to look at some possible sources of variation that can occur. One is geographic variation. Now, geographic variation includes environmental factors. So animals that live in warmer climates are going to have adaptations to live in warmer climates. Animals in colder climates are going to have those adaptations as well. So like a whale, for example, has a lot of blubber, allows it to live in colder climates versus something in the desert like a camel. Camel's got the ability to retain water that it needs if it lives in the deserts. Something else that's geographically related to is something called a cline. It's basically a graded change that has along some transect that you can actually see speciation occur across those 2 different transects. So here's an example of one. These are step clines, in a rat snake. So you can see all the different locations in which you would have different rat snakes. They all come from the same ancestor, but there's just areas in which you're going to see that variation. So even though color patterns can change on the snakes, and there's actually no environmental cause for those changes. Another one is actually altitude. So here you have the Sierra Nevada range, and then you have that basin plateau, and you'll notice that as the altitude increases, what happens to the plants? They get shorter, and that's because there's less oxygen that's available up there, so they don't respire as quickly, therefore they won't have the ability to photosynthesize as quickly either. Another example are these mumachog fish. They vary in a cold adaptive allele, which is this LDHB allele frequency. So in areas where you have much cooler climates, you're going to see a much higher frequency of the allele, almost close to 1. And then as you go down in latitude towards warmer waters, you're going to see much, much, much lower frequency of those cold adaptive alleles because they don't need them in those warmer climates. This gene specifically codes for an enzyme that allows them to live in those cold environments. So of course, you're not going to need it as much in those warmer environments. The other types of variation that we've talked about before, of course, mutation. We have point mutations, frame shift mutations, or chromosomal mutations. We also have rapid reproduction. There are some organisms that reproduce in a couple of days. Fruit flies, for example. Prokaryotic viruses, very fitting for this particular, prokaryotes and viruses, I shouldn't have said that. Prokaryotes and viruses, again, very fitting for what's going on right now. And then sexual recombination. So again, we've talked about the fact that when sperm meets egg, there is no rhyme or reason to which sperm meets the egg. So you're going to have that variation that occurs there. Let me pause this. Let's skip over to a different slide. We've already talked about microevolution. Forgot about that. Here we go. This is the last thing I want to talk about. These are balanced polymorphisms. And polymorphism is the ability of natural selection to maintain diversity in a population. So we're looking at balancing selection. So when you do your POGIL tomorrow, you're going to look at the 3 different types of selection. You have disruptive and balancing and stabilizing. Stabilizing and balancing are the same thing. Directional, disruptive, and balancing or stabilizing. In stabilizing selection, you usually preserve the average, and the curve will start to actually steepen so that you preserve more of that middle of the road or average variation. So there's two methods of preserving variation: heterozygote advantage and frequency dependent selection. So heterozygote advantage, basically, if you are heterozygous for a certain trait, if that allows you to survive better in a population, then natural selection will occur, and those who are heterozygous will actually survive. They have more reproductive success. We've talked about this before with that fitness. So heterozygotes have a higher fitness than other genotypes would. An example of that is sickle cell anemia. We didn't get a chance to do one of my favorite activities with the cards last week to really see how heterozygotes have an advantage. So remember, let's think back to sickle cell anemia. Blood cells become sickled under low oxygen conditions. So in the capillaries, there's blocked blood flow, causing damage to the kidneys, the heart, brain. Any of those symptoms could occur, especially mostly under low oxygen stress. But if you have both alleles for sickle cell anemia, those symptoms can actually manifest more frequently than others. So in heterozygotes, you have, so basically there's different alleles. So you have the normal blood allele, normal hemoglobin allele, you have the sickle allele, and if you are heterozygous, you actually have one copy of each. We're looking at codominance here, in effect. So if you're heterozygous, you have some sickled blood cells, but others are mostly normal, and under low oxygen stress, you would have more of those sickled cells be affected. Here's a really interesting thing. So you'll notice on the graph that we're looking at Africa as a whole, and you're going to see frequencies of the sickle cell allele. So you're going to see areas where you have a much higher frequency of the allele, and then you're going to see frequencies where you have a lower, or areas where you have lower frequency of the allele. You'll also notice that there is the distribution of malaria caused by the protozoa that lives in mosquitoes, and that is represented by the dots everywhere. So in areas where you don't have that mosquito, you're not really going to see the sickle cell allele. You can see that those are the white areas, or it's going to be much lower. And then in areas where you have a higher frequency of the sickle cell allele, you will notice that it corresponds to areas where malaria is prevalent. So people who are heterozygous actually are at an advantage, because if you have sickle cell anemia, meaning that you have both copies of the sickle allele, you will eventually not survive because of the sickle cell disease. If you are homozygous dominant, meaning that you have both of the normal hemoglobin alleles, you have a much higher chance of getting malaria and dying from malaria. However, if you are heterozygote, you actually have some of those sickled cells which don't allow the malaria protozoan to live in those blood cells. Therefore, you will not get malaria, and you will not be affected by malaria, and you won't be affected by sickle cell. So that advantage remains, which is why the sickle allele remains within the population. Here's a second type of selection that we're going to look at. This is frequency dependent. This is where rarer phenotypes are maintained. Most common phenotypes become eliminated and decrease in number. So the example here is the African swallowtail butterfly. So this is our disruptive selection. There is this butterfly that mimics poisonous butterflies. So it looks like what we call the mocker swallowtail. And there's a specific banding pattern that intermediates are selected against. Okay, so the ones that are in the intermediate phenotype, meaning kind of in the middle, like I talked about before, they don't look like the poisonous butterfly. Therefore, they're the ones that are going to be eaten versus the ones that are on the other extremes, which is why we call it disruptive, because you have the different extremes that are being selected for, because the intermediate is going to decrease in number because they're being selected against. Here's another example. These are scale-eating fish. So you see that you have the left mouth and the right mouth. Excuse me. These are scale-eating fish in Africa. So they actually attack other fish, which is the red guy in the middle, from behind. And you'll notice that there's the right mouth and the left mouth. So these guys are really weird. So they attack from behind, they dart up, and they actually eat the scales off of the fish. Um, right mouth is dominant, and what it does is the right mouth kind of twists. So if you have your right hand, this is my right hand, so we'll do this one, and you go like this, it twists this way. You can't, well, you guys you can, but go like this with your mouth, and we twist it like this or twist it like this. I know it's kind of weird to think about, but these right mouth fish will come up on the left side of the prey and eat the scales off the left side. So what happens is that the fish start to realize, oh, the my predator is going to come up on my left side, um, which then they know, hey, I'm going to continue to check out my left side. So what happens is those left mouth fish actually have the advantage because they're not, their prey is not looking the way that the left mouth fish can eat. So what happens then is that the left mouth fish become more dominant, and then the prey start to realize, oh hey, now I have more fish on my right side. So then they're going to start looking to the right side. So then the right mouth fish are going to have an advantage because they're going to become more dominant. So this actually goes up and down and up and down and up and down, and the frequency changes. It evens out at about 0.5 between both the right mouth and the left mouth, but you can see that fluctuation between 1981 to 1990. So just a 10-year span where it kind of fluctuates up and down. So it oscillates over time. So that's it for me. I hope that this helps, and please let me know if you have any questions, and have a great rest of your day.